So as folks begin to make their way into the webinar, we'll wait a few minutes to get a few more folks in here, and then we will kick things off. So sit tight, be patient for a couple minutes, and we'll get rolling soon. All right, let's start to get things underway as the last few folks make their way in. On behalf of the BioMaker space and the BioMakers group, I'd like to welcome everybody today. Joining us, this is the last of our summer installment of the COVID-19 BioMaking Solutions virtual seminar series. Our speaker today is the 10th in our series, which you can see up on the screen. We've featured a really wide range of innovative approaches to combating COVID-19, and it's been a fantastic series of leaders from both academia and industry. We've heard about new approaches to diagnostics for individuals as well as for community level epidemiology, novel and diverse approaches to vaccine development, computational and clinical efforts toward therapeutics, and tools for accelerating and scaling the commercial production and deployment of these types of solutions. So I'd like to just thank all of our speakers for making the time from the busy schedule, especially during these challenging times, to make this whole series possible. And to thank all of our participants over these 10 sessions as well. They've been well attended and well received. So appreciate the good questions from the audience, and we'll start to launch into this one. A couple quick notes before we begin. Please do hold all the questions till the end of the talk. When you have a question on the participants tab, you can use the raise hand icon. I will enable you to come off mute. You can ask your question directly to our speaker. If you do have any trouble, reach out to me. I'd be happy to assist you. We are recording for today, and we'll make a video recording available as soon as that can be reviewed and edited. So I have the pleasure today to introduce our speaker, Dr. Brendan Manning. He has received his bachelor's and PhD degree in nanotechnology from University College in Dublin. His PhD thesis was on photopatterning of surfaces using photolyable hairpin oligonucleotides. From there, he continued into a postdoc at CSIC in Spain, working further in surface patterning and DNA origami with Professor Ramon Irita. After which he moved into some in vitro diagnostic development in industry as senior scientist at BioKit. And then from there came over to the United States and joined T2 Biosystems as a senior development scientist, where he moved into various management roles in product development for molecular diagnostics, while simultaneously developing his business schools through Harvard Business School program. He joined SHERLOCK Biosciences about a year ago as the Associate Director of Assay Development. So welcome, Dr. Manning.
Thank you, Justin. And thanks for the opportunity to give this talk today. All right, I hope you can see my screen.
Looks good.
All right, great. So I'm going to start with a little bit of company background. So SHERLOCK Biosciences is an engineering biology company that is focused on leveraging CRISPR and synthetic biology to develop breakthrough platforms that enable rapid, low-cost diagnostics. These diagnostics tools will improve healthcare by expanding the availability of molecular diagnostic testing. The company was founded by a team of pioneers in the fields of CRISPR and synthetic biology in 2018. So what do we mean by an engineering biology company? We have two core platforms. Inspector, and SHERLOCK that are based around a series of technologies such as CRISPR and cell-free expression systems. Our aim is to leverage these platforms to enable and accelerate solutions that will generate new and impactful data that can both evolve the platform and improve healthcare for all. So that's the mission of the company. Our two platforms can be broadly distinguished by function. In SHERLOCK, a target nucleic acid activates the functionality of a CRISPR guide complex, causing cleavage of a reporter. And in Inspector, the target generates a pre-programmed functionality, such as an enzyme or a polypeptide. These platforms have advantages that we are leveraging both individually and combined. SHERLOCK is both highly sensitive and specific, robust to minimal sample preparation, and is the first CRISPR technology FDA authorized for clinical use. Inspector is a programmable synthetic biology-based platform that involves amplification-free molecular detection at ambient temperature and is compatible with simple, affordable readout methods. Next, we will move on to talk about the impressive research that is the foundation of the SHERLOCK CRISPR SARS-CoV-2 assay. SHERLOCK has been published in Science 4 times, each time advancing the system to the robust state it is in today. In the initial publications, the SHERLOCK technology presented the value of collateral cleavage for the detection and was then designed to be more sensitive through sample amplification, which was finally demonstrated to be multiplexable, quantifiable, and was combined with a simple lateral flow readout that was deployed on real-world samples. Here we have data from our co-founder, Feng Zhang's lab, which demonstrated the high sensitivity of the SHERLOCK method. In this case, detecting a low concentration of a target down to a single molecule per sample level. The process begins with the recombinase polymerase amplification step. The amplified target then binds to a specific Cas guide complex, which results in collateral cleavage of a fluorophore linked to a quencher. This fluorescence readout can then be detected on a plate reader. In these early SHERLOCK publications, the high specificity of the diagnostic platform was also shown by demonstrating that a variety of bacterial and viral pathogens could be distinguished both accurately and reliably. In this example, single nucleotide specificity was demonstrated by changing the CRISPR guide sequence so one could distinguish between a wild type and mutant allele of a gene associated with cancer. Another advantage of the SHERLOCK method is the robustness of the process to sample type. In this publication from our co-founder Pardi Sabeti's lab, her team developed a simple method to deactivate endogenous nucleases present in a sample and access viral nucleic acids that enable the detection of a target by SHERLOCK. The same team also demonstrated that this sample processing method can be applied to a wide variety of common sample matrices, such as saliva, urine, and blood. This method, combined with SHERLOCK, was successfully used to detect viral, bacterial, and human targets in clinical samples, which really demonstrates both the robustness of SHERLOCK to sample type and to sample extraction. SHERLOCK can also be adapted to a variety of readout methods. As I mentioned earlier, SHERLOCK was initially developed with a fluorescent readout that involved the cleavage of a fluorophore bound to a quencher. Since these initial efforts, the platform has been coupled to a simple paper-based readout, similar to a pregnancy test, but that can be applied to a viral target such as SARS-CoV-2. Recently, SHERLOCK was adapted for massive multiplexing, using a fluidic chip known as Carmen that enables one to run thousands of SHERLOCK reactions on one chip. Using this method, it was possible to test for all 169 human-associated viruses at one time. This technology opens up a broad range of opportunities for using the SHERLOCK assay to detect many pathogens from many samples simultaneously. This technology can be used for high throughput screening and pathogen surveillance. In summary, the SHERLOCK technology is a powerful new nucleic acid detection system that is both ultra-sensitive and ultra-specific, capable of single molecule detection and single nucleotide discrimination. It is compatible with simple sample preparation methods and adaptable to multiple readout methodologies. So in the next section, we'll discuss in detail a little bit of the development of the SHERLOCK CRISPR SARS-CoV-2 assay. To begin with, this is what a typical diagnostic development process might look like. As you can see, we have discussed the foundational research, which is really the starting point of the assay development process. From this point, you have a sense of the technological capabilities of a platform and what the customer will want in any given product. Moving forward into design inputs, you translate these user needs into design requirements for your assay while optimizing and further developing the product. You then end up with a set of design specifications and scale up of manufacturing lots of materials. The next phase is focus on design verification, confirming the product meets the design specifications, and what's known as design validation, which is focused on confirming that the customer needs are consistently met. And finally, you arrive at a commercial product. In a normal world, this would normally take 2 to 5 years and likely somewhere on the longer end for a new technology. This is what makes what the team at SHERLOCK Biosciences has achieved in the last year so remarkable. In the less than three months, we were able to pivot and rapidly develop an assay to detect SARS-CoV-2. So on New Year's Eve 2019, the first reports of a pneumonia cluster in Wuhan were published. By January 21st, the first case was reported in the US. Retrospectively, there were likely a handful of cases in the US at this time. In early February, we ran our first SARS-CoV-2 SHERLOCK assay in-house. On March 10th, we received viral RNA from the University of Maryland, Baltimore. This was really a turning point in our assay development and gave us the confidence to move forward. Prior to this, we'd been using synthetic RNA. By April 25th, we had submitted our EUA data package to FDA, and the emergency use authorization was approved by FDA on May 6th. So I'll walk you through the process of our SHERLOCK CRISPR SARS-CoV-2 kit. The process starts with extraction of RNA from an upper respiratory tract sample. Target nucleic acid sequences are amplified by reverse transcriptase loop-mediated isothermal amplification. That amplified target nucleic acid is specifically detected by a CRISPR-Cas complex, which consists of a Cas13 enzyme complexed to an RNA guide. When the CRISPR-Cas complex binds a target, it activates collateral cleavage activity that will cleave a fluorescent reporter, resulting in a fluorescent output. As previously described, the SHERLOCK platform is compatible with multiple amplification technologies. The SARS-CoV-2 kit leverages RT-LAMP. After an initial reverse transcription step that converts viral RNA to DNA, the DNA is amplified by DNA polymerase with strand displacement activity. One of the advantages of this method is that it is an isothermal process that occurs at 61C and can be carried out on a simple heat block. The kit detects 3 targets, 2 SARS-CoV-2 specific targets for the ORF1ab and nucleocapsid genes, and a human control, the RNase P POP7 gene. On May 6th of this year, the FDA granted emergency use authorization of the SHERLOCK CRISPR SARS-CoV-2 kit. This was the first authorization of CRISPR for clinical use and an important milestone for SHERLOCK Biosciences and for the use of CRISPR technology in the clinic. The intended use of the SHERLOCK CRISPR SARS-CoV-2 kit is for the qualitative detection of SARS-CoV-2 in upper respiratory tract samples and bronchoalveolar lavage specimens from individuals suspected of COVID-19. Sample types approved include nasal swabs, nasopharyngeal swabs, oropharyngeal swabs, nasal aspirates, and BAL samples. The assay can be performed by qualified and trained clinical personnel in a high complexity laboratory. The limit of detection is 6.75 copies per microliter of target in BTM, and sensitivity and specificity with contrived samples were both 100%. The time to result is approximately 1 hour post-extraction. The extraction method approved is a pure-length viral RNA DNA mini-kit from Thermo Fisher. The required instrumentation includes a Biotech Neo 2 plate reader running Gen 5 3.08 software. The limit of detection was determined to be the lowest concentration at which greater than or equal to 95% of the samples are positive for a minimum of 20 replicates. Samples were prepared by pooling negative patient samples and spiking with quantified viral RNA. The limit of detection for both SARS-CoV-2 targets was determined independently, with the limit of detection for the ORF1ab target determined to be 6.75 copies per microliter, and the limit of detection for the nucleocapsid target determined to be 1.35 copies per microliter, which is right around the range of a qPCR assay. An in silico analysis was performed to determine the inclusivity of the SHERLOCK CRISPR SARS-CoV-2 kit and involved analysis of over 9,000 individual strains of SARS-CoV-2. These were aligned with all primer and guide sequences from the ORF1ab and N targets, and mismatches were identified. No genome was identified with a mismatch in both targets, and all known sequences of SARS-CoV-2 would be detected by at least one of the target tests. And this was probably one of our biggest challenge. At the beginning, when we started designing our primers and probes, there was a handful of genome sequenced, and really there's been an exponential growth in the data of the sequencing data out there. So it's well over 9,000 strains now, and periodically we go back to confirm our inclusivity and exclusivity. Analytical specificity was also evaluated in silico and by wet testing. The in silico analysis of the nucleocapsid and ORF1ab primers and CRISPR RNAs designed for these SARS-CoV-2 targets showed no significant similarities to the organisms listed. We also performed wet lab testing on a subset of the organisms listed. No cross-reactivity or competitive inhibition was observed in testing with any organism. This testing focused on pathogens common in upper respiratory tract samples, such as the 4 common human coronaviruses in the top left of the table, influenza A and B, and staph epidermis. Clinical evaluation was performed with contrived clinical samples due to the lack of availability of clinical samples at the time of running this experiment. Individual nasopharyngeal swab samples confirmed to be negative for COVID-19 were used unaltered as negative samples or spiked with viral RNA at 2, 3, or 5x the LOD of the assay. Positive percent agreement and negative percent agreement were both 100%. We have also worked with clinical partners who have run patient samples with the SHERLOCK CRISPR SARS-CoV-2 kit, and they are also reporting very high concordance with both positive and negative samples. The SHERLOCK CRISPR SARS-CoV-2 kit is the first FDA-authorized use of CRISPR in the clinic and has many benefits, including rapid time to result, high sensitivity and specificity, as demonstrated with our EUA clinical evaluations. Importantly, the materials in the test have a unique supply chain when compared to qPCR tests, and the test can be run with equipment and instrumentation that is both common and not used for standard qPCR assays. That concludes the section on the SHERLOCK CRISPR SARS-CoV-2 kit. Now I'd like to spend a little bit of time talking about SHERLOCK Biosciences amplification-free synthetic biology-based platform called Inspector. Much like the SHERLOCK platform, Inspector targets nucleic acids in saliva, urine, blood, or other common sample matrices. The process involves a nucleic acid target present in a sample that is hybridized to two probes, which are then fused and polymerized to form an expression cassette. This expression cassette is then used as a template for a cell-free expression system, and via transcription and translation, a pre-programmed output can be produced. This output can be an enzyme or a polypeptide. The real power of the Inspector platform is that it allows us to move towards home testing of nucleic acids. This will help us bridge the gap from large, automated, high-throughput instruments, low-throughput point-of-care systems, to a test that's much more accessible and much simpler, much like a pregnancy test. The advantages of a system like this are now more apparent than ever. To summarize, I have presented two novel, unique, and powerful molecular diagnostics platforms. SHERLOCK has high sensitivity and specificity, is compatible with existing instrumentation, and can be used in today's healthcare environment. Inspector has high sensitivity, does not require instrumentation, and can be performed at the point of need, the home, and in low resource settings. These platforms have already been applied to a variety of applications, including the detection of SARS-CoV-2. So that concludes my presentation. I think we have time for some questions. Thank you.
All right, thank you very much. So as I mentioned at the beginning, if you would like to ask your question out loud on the participants tab, you can use a raise hand icon, and that enables me to bring you off of mute to allow you to speak directly. Find that is most effective in making sure we have good communication, but if that doesn't work for some reason, I can take questions in other ways. All right, good. We got Stacy asking a question. You should be able to unmute yourself and ask now.
Okay, hi, thank you for a nice talk. I was curious about the supply chain for the enzymes required for this assay, and what that's looking like. Are you in conversations upstream to secure that supply chain for broad implementation, particularly for SARS-CoV-2? Thank you.
Yeah, that's a good question. So for Cas13, which is the CRISPR Cas-based enzyme, we have the rights for that as a diagnostic tool. So we have multiple suppliers of that. Right now we have IDT supplying the kit, and they're obviously a huge manufacturer and can handle scaling and manufacturing at incredibly high volumes. The amplification side is LAMP, which we're sourcing from NEB right now, and also a very large company which hasn't had problems with producing that enzyme. The nice thing is that, as I said, these supply chains are very distinct from what is traditionally used in qPCR, with the exception of primers and guides. But thankfully, there hasn't been much of a problem accessing primers and guides yet. So hopefully that doesn't become an issue.
Thank you.
All right, well, as other people continue to think of and prepare questions, I was curious if you could tell us a little bit more about the selection on the targeting sequences, if it was just based on alignment and the degree of predicted specificity, or if there were any other key drivers for which regions were selected as the targets.
That's a good question, and we you know, we discussed it a lot. We also designed a lot of targets. We, we, we kind of had a lot of talk about it, and then we decided to brute force it and just develop everything we could. So we designed, I think it might be 80 different targets. And we did look at genomes of nearest neighbors and tried to remain, you know, inclusive of things like bat SARS. But, you know, when we were looking at this target, it was January 2019 or January 2020. So there was, you know, I think it was less than 10 sequences available to us. So we really had no idea. I mean, you know, we knew it was going to mutate, and it was going to mutate every week. So, you know, that was one of the reasons for putting in multiple targets into the assay. I think it's kind of risky to just have a single target, and having two distinct targets and two distinct primer sets in two distinct regions, I think helps with inclusivity. So it's definitely a challenge for us, and we periodically went back and kind of double checked that things were still okay. And we did have some redundancies in targets that we could, you know, we had optionality that if something, you know, ended up not having the required inclusivity, we could swap over to another target.
Sounds like a good approach. We have a question from Melody. Melody, you can unmute and ask if you'd like.
Sure. I was wondering how you imagine the test will scale in terms of getting to people's homes, workplaces, et cetera, for testing, because I imagine that being the biggest kind of bottleneck currently.
Yeah, that's a good question. I think it's a complex problem. You know, there needs to be increases in capacities at kind of all levels of the process, including, you know, access to sample collection devices, shipping of samples, and then processing tests. And that seems to vary regionally. You know, we're in constant communication with our partners to make sure that, you know, from our side, we can supply the reagents and the kits that we need. But it's a complex supply chain that seems to be, there's a lot of pressure on it right now. So I think it'll take some time before, you know, that all gets figured out.
Thank you.
All right. We have a question from Karthika. You should be able to unmute.
Yeah, I was just curious, how far are you down the lane with the inspector-based kit for SARS-CoV-2? Like, what is the time frame that we're looking at?
Yeah, we're working hard on that right now. We haven't announced any time frame, so I'm not going to announce one here. But it's a technology platform that is at an earlier stage than SHERLOCK. But it's obviously, you know, it's crystallized that it's become, you know, much more necessary. So we're trying to accelerate it as much as we can and as fast as we can, because we definitely see that it has, you know, huge value to the current pandemic, and could really answer, you know, a lot of the problems that people are having with testing. So we're working as hard as we can on it, and we hopefully can have a solution as quickly as possible.
Also, I have one more question. So are you like thinking of developing the inspector-based kits for other flu-like viruses, or are you just focusing on SARS-CoV-2 for now?
So right now we're focused on SARS-CoV-2 as our main target of interest. But there's definitely a lot of interest in parsing out influenza A and B and SARS-CoV-2, especially as we move into flu season, and also, you know, other diseases like RSV and other common coronaviruses. So definitely we see value in the ability to detect multiple pathogens from a sample. But our focus right now is SARS-CoV-2.
Okay, thank you. Thank you, Brandon.
Thanks.
And looks like we have another question from Stacy.
Hi, just a question about the kind of the multiplexing and ability to utilize.
Oh, sorry, that would have been my mistake on muting. If you can repeat on that, sorry.
Sure, no problem. I'm not sure what you heard, but let me just start from the beginning. So I'm curious about the platform capability around untargeted detection of multiple viruses, for instance. So looking for new viruses or things that you might not know exist, and how that impacts the limit of detection when you go to those untargeted approaches.
Yeah, the challenge with untargeted approaches is going to be sensitivity and specificity, and the fact that, on a molecular level, you don't really know what you're looking for. So, and for viruses, that's particularly challenging because of the number of mutations and SNPs you have in every single virus. So even just detecting all of the strains of influenza A and B out there is a huge challenge. Trying to predict what the next, cause of a pandemic or the next seasonal flu is going to be is somewhat challenging. So in terms of untargeted approaches, that's something that's certainly a challenge. It can be achieved by NGS sequencing, but obviously, that's very, very expensive to do on a kind of a population-wide scale. So, SHERLOCK can be applied to kind of massive multiplexing, as we've seen from the Karman system. The way that works is kind of a non-specific amplification-based process. For coronaviruses, you could definitely do that in such a way so you're able to detect all of the known coronaviruses, which would give you some idea of, if there's a new pathogen or a new strain that, has jumped species, for example, which seems to be what happened with coronavirus. So if you're detecting a common strain of bat coronavirus in humans, you probably know that something strange is happening. But that's a big question, or that's a big challenge for epidemiologists, and kind of sequencing is the de facto standard. I think the Karman system, with its massive multiplexing, has the potential to really help in that area.
Thank you, Brendan.
So, Brendan, I know you weren't able to talk too much to the inspector timeline, but is there a summary of target specifications and what you're hoping to accomplish with that? Things like, time for the test to develop and to get a reading, price point, again, sensitivity, limit of detection types of metrics there.
Yeah, I think, in terms of price point, where, ideally it would be, somewhere under $10, I think is probably the price point you would ideally want. It'll be based on a lateral flow readout. So that's, the standard there is relatively cheap. Our hope is the sensitivity and specificity are close to qPCR, if not between qPCR and immunoassays. Sensitivity and specificity for the rapid immunoassays tends to be a little bit lower. So hopefully, targeting qPCR, but potentially somewhere between the two. And then, yeah, we have a set of target specifications that we're kind of looking at. I think, inclusivity, broad inclusivity is probably the most important one, to make sure you're not missing any significant clades of coronavirus, and high specificity, which seems to have plagued a lot of the rapid antigen and antibody tests out there. Time to result, if you ask somebody in marketing and business, they want it in 5 minutes or as fast as possible. And obviously, we're going to do everything as we can to make it as quick and as rapid as is possible. But obviously, we're, right now we're going to be pushing to have something out as quickly as possible. I think that's the most important thing, irrespective of time to result, especially if you can make it an at-home test. There's probably some more tolerance there for a slower test versus, the rapid point of care tests, for example.
Mm-hmm. And target fluid for that? Were you thinking of saliva? I'm sorry if you mentioned that, or a nasal swab and self-administration of that.
Anything at home would have to be self-collected, so it has to be a nasal swab or a saliva sample. And if you look at the qPCR data for saliva samples, sensitivity seems to be relatively high, and viral load seems to be relatively high in saliva samples. Especially when you look at the disease progression, concentrations seem to skyrocket very quickly and then kind of trail off pretty slowly. So saliva or a nasal swab would seem to be the obvious sample of choice.
Looks like we have another question from Karthika.
Yeah, I had a quick question. So if you're looking at saliva samples, are you just thinking of just spitting on the lateral flow tip, or would it require like pre-processing of the sample?
So as the process stands right now, it would go through a processing step. Our hope is to integrate that into the lateral flow system. So, I don't know if you guys are aware of, the saliva collection tubes for 23andMe and things like that, where, the saliva is aliquoted into a tube, and then there's some solution mixing that's kind of automated into the tube. There are some lateral flow systems out there that are kind of similar to that, which we're evaluating. And then there's lateral flow kits that are kind of integrated that will do some fluid manipulation. Really the target with any at-home test is that the user cannot really be expected to do anything, right? It all has to be automated, simple, and error-free. So that's the kind of design principles we're working on. So from a user perspective, you would, donate your sample and then, with some simple manipulation, walk away, come back, and then look at the test results.
Thank you.
We still definitely have a little bit more time if folks do have more questions. Let's see. So Brendan, something that we've heard frequently or commonly in the diagnostics and the testing are people asking questions about pooling or multiplexing on samples as a way of getting more throughput, reducing costs. Is that something that you've been thinking about? Is that a benefit within this, or would the target be to go one person, one test? Oh, you're still on mute. Sorry.
Sorry, pooling, is a great strategy up until, your prevalence gets too high. So it's really only viable up to, I think, a prevalence of 7 or 8%. And then you end up just having to reflex test too much that, the time to result gets so low that it doesn't really help a whole lot. So yeah, we're certainly evaluating how to push throughput. We're working on, automation and ways to automate the process. The nice thing is, in principle, the process is very, very simple. It involves very few pipetting steps. So it can be automated, and with the amplification step carried out off instrument, basically, you're amplifying on a heat plate or any kind of thermal block. But you're reading on a fluorescent plate reader, you can kind of stack up and do multiple 384 well plates at one time, and then read every 10 minutes. So you can get a lot of throughput by optimizing the automation side of it. Whereas most qPCRs, you prep your plate and then you leave it on the instrument for 2 hours. Whereas you only get a result after an hour. So there's an hour of dead time there that isn't really valuable. So we're working, both on pooling to improve throughput, but also on, ways to optimize the assay and to optimize the workflow to enable higher throughput.
Sounds good. Jantine, would you like to ask your question?
So this was incredibly interesting. So I've been really interested listening. Maybe Brendan, I'm a philosopher ethicist at the Wyss Institute, and my specialty is in synthetic biology. So I'm sort of familiar with things like CRISPR and heard about SHERLOCK and Inspector early on. I would want to ask, have you as a company been thinking about, and do you have any, plans or even at the level of ideas in place about how to, distribute this in an equitable and just manner? Because it's clear there's some places there are bigger needs than at other, but also unfortunately at the places, even in the US society, but also globally, the places where it's most needed are the least able to afford such things. So have you been thinking about it? Have you been considering this?
Yeah, I think as a company, really, that's one of our fundamental goals. Prior to the pandemic, as a company, we're striving to make molecular diagnostic systems that are really available to everyone at a price point that anyone can afford. So it's kind of, it's somewhat ingrained in everybody we hire, that's a goal. Both SHERLOCK and Inspector, I think, move the needle in that way. It's debatable whether we're moving fast enough, but we're trying as fast as we can. I think a product like Inspector, we're designing it for low cost. I think when you build that philosophy into the design process, I think it helps drive down cost, and you're always cognizant of it. And you're always aware of how much your reagents and your consumables and all of the components that go into a test cost. So that's one thing we're really focused on designing in, to try and make these assays as cheap as possible, which we hope will go some way to enable equitable distribution, both in the US and outside of the US. But I think it's that's a big and difficult question. I hope I answered it adequately.
Yeah, no, that's, and I think early on with like Ebola, it was sort of clear, but it was also from, people where the place where it happens, they need to be able to use tests, any tests that are technically suitable and that are affordable. But with Ebola, to some way, it was like, say, comfortable for us here in the Western world. Like, well, this was far away, we wouldn't get it. And now with the SARS-CoV-2, the situation is completely different. But we see also in our societies really the painful places where it would be needed. As to the, but because it's also a global problem, how about with Inspector? Because you mentioned ambient temperature, how about say like tropical areas or maybe very extremely cold areas? Are there temperature requirements that would put restrictions on availability?
Yeah, so we're still in the development phase, but we are designing for ambient temperature. Exactly what those temperature ranges will be, I can't really say right now. But the standard, I think, from the WHO on ambient temperature is something like 10 C to 40 C. I think that's their target, which is that's a challenge. That's a wide temperature range, but usually brings you into basically anywhere there's people, you can run it. So it's definitely something we're trying to design and develop in. And the other thing is reagent stability and trying to not require cold chain storage, because that just reduces the kind of the places that you can send tests. So we're hoping it's a device that both works in ambient temperature and is stable without cold storage.
Yeah, great. Thank you. Thank you. That was very, very helpful and informative. Thank you very much.
Thank you.
So let's see if there are any other questions that come up. But I was wondering if you would tell us a little bit about your motivation on your career path and trajectory. Kind of what drove you to get into diagnostics?
Yeah, that's a good question. I always like DNA and RNA, so that's kind of at least my PhD and my research background. I've always been kind of fascinated by nucleic acids. I see you maybe are too, by your biomaker in the background, your nucleic acid on your B. So I guess that's something that's always interested me. And then diagnostics just it just was an easy fit for me. It's always nice to do something that's rewarding and is both challenging and rewarding, right? It's nice to see your research turn into products that then are tested on people and can go some way to help people. I kind of this whole SARS-CoV-2 outbreak has probably been the nicest example of that, right? It's the first time I can tell my parents exactly what I did and point to it and say, hey, this is the job that that we do and how we're trying to make a difference.
Yeah, and kind of in a relative spectrum of importance, tell me about kind of gaining more of the business background and insight, and how you have felt that be integrated into your day-to-day role. And well, I can give my position up, but I'd love to hear your side.
Yeah, I guess when you start as an academic, you're basically focused on research and what's possible, which is super interesting, right? Like trying to make things work and trying to make them possible and see how how far you can push the envelope. Kind of what I learned as I transitioned into industry was that there are these things called user needs and product requirements, and they really drive the research that we do here. There is no point doing research on something that's not a viable product or doesn't meet the need of a user. So that's really driven me to kind of understand more about where these things come from, and they come from discussing having conversations with key opinion leaders, with patients, with advocacy groups, and understanding, okay, what is the real need and how are you going to use a product? And there's a lot of challenges associated with that, which I think, I underestimated when I started in diagnostics. It is not easy to figure out what a product should do and then what the trade-offs should be, because all products have a trade-off, right? Whether it be sensitivity, specificity, time to result, there's always trade-offs. So understanding, what the right trade-offs to make is a very difficult decision to make. So really that's kind of been my driver moving more towards business is understanding where those decisions are coming from and where that, the market research is leading, and how do you look at market research as opposed to, fluorescence values or qPCR data, things that I'm comfortable with. So it's another data set which drives everything we do.
Yeah, absolutely. I agree completely. I think that's very well said. I hope we have some folks in the audience who are planning on taking 2380 this fall. And if so, listen and pay attention, because that's very good advice toward your design projects. And I may have to ask you to come to a class session and tell the students about that as well.
That'd be great. Yeah, I'd like that.
Well, it looks like we've gotten through about all the questions that folks have. So we can probably wrap up a bit early today and give everybody a little bit of time back. So just on behalf of all the attendees, I'd like to thank you for coming and telling us about the work that you're doing with SHERLOCK Inspector over over at Sherlock Biosciences.
Thank you. Thanks, everyone.
And everyone, have a great rest of your afternoon, and we will let you know upcoming programming from the BioMaker space. So, goodbye.